Well, welcome everybody. This is our November 14th meeting of the GLIMPRINT and the IMAG-MSM Working Group on Multiscale Modeling and Viral Pandemics. I'm very pleased to see everyone here. Thank you for coming. As always, I need to remind you that this meeting is live streamed and recorded and made available on YouTube for later viewing in a slightly edited form. The viral pandemic seminars page is back up after a snafu at NIH. We thank Jim Sluka very much for getting it back, all of our content back after a bit of a nervous period. Please, if you notice any issues with it, missing material, please email jsluka@iu.edu. As always, Tom Helikar and I and Jim and Lorenzo are available to take your comments, suggestions, criticisms. Please do let us know what we can do to make this community more useful and vital for you. We have our usual communication channels, some of which get used more than others. There's a Slack channel, which doesn't get used very much. There is a Twitter X handle, MSM Viral. There is the IMAG MSM Wiki page. There is the IMAG LinkedIn page, and there is YouTube. And there's the GLIMPRINT page as well, which Jim has been working on along with other people in the community. If you'd like to help us develop the web pages, we would be more than happy to have help with the web presence and social media. If you can retweet the weekly announcements and use that MSM Viral handle, we would be most grateful. It would certainly help build community attendance and awareness. If you have a publication to share related to this community, we can put it up on the IMAG MSM page and also on the GLIMPRINT page. We want to make sure that everybody doing work in this field gets the attention they deserve. And so please send any of content that you would like to see put up to jsluka@iu.edu, and he'll do that. The GLIMPRINT page can post job announcements, conference announcements, and so on. If you would like to do that, there is a form online. Submit to GLIMPRINT News. We'll do our best to get your word out there. Again, jsluka@iu.edu if you have any questions about that. If you find any problems with the web page, please let us know. We're always a volunteer organization, and it's always a work in progress. Reinhardt is still recruiting in Florida, and Tom Helikar is recruiting in Nebraska. And so both positions have multiple positions available. Please contact Reinhardt or Tom if you're interested in either of these. They're both great opportunities, maybe for you, maybe for your students or your postdocs. Please do get in touch with them if they're of interest to you. There is the ARPA-H Catalyst program for computational toxicology and physiology coming up. There is a teaming page there where you can, I believe, still post if you're interested. It's quite an interesting program. It's quite well funded. It can include Europeans as well as Americans. It's not limited to the US. It's an international program. And so you may want to look at that. There are some new NSF announcements in the digital twin area, which you might want to look into. These are awarded grants rather than grant opportunities, but it may give you ideas. There is the January 14th deadline for the NSF POSE, Open Source Ecosystems program. That is limited to the US, but may be of interest to many people here who develop software technologies. There is the NIH NOSI, Systems Modeling of Infection and Immunity Across Biological Scales. We missed the October 5th deadline, but those are on the regular 3 times a year deadline schedule. And so many people I know have submitted or are planning to submit for that. That one, again, can include an international component. We've been showing this for some time. A year from now, there's going to be a meeting on virus dynamics in Bordeaux, France. Again, if you have conferences or meetings that you would like advertised, please do let us know and we'll try to get the word out to the community. I just heard today, Reinhardt has known about it, I guess, for some time. There is a NATO-sponsored meeting on human digital twins in the military, December 10th through 12th in Florida. And you can register for this up till the 22nd of November. And so if people are interested, that is possible. It's in person only. It is open to anyone who is a member of a NATO country. So that would be most of our European colleagues. And as they also list a few additional countries that are eligible. I realize it's a little bit late to be planning to go to Orlando on the 10th to 12th of December, but I think this could be quite interesting for many of us. Upcoming seminars. In 2 weeks, we will not have a seminar. It's Thanksgiving Day in the United States. We will meet on December 12th. We won't meet on December 26th, Boxing Day. And so then we will resume seminars in January. January 9th is our first scheduled seminar with a European speaker. We have openings for the 23rd of January and the 20th of February. As always, you've heard me say it now hundreds of times, literally. Please suggest your students, your postdocs, yourself, the people that you would like to hear from, and we will do our very best to get them on the schedule. You can send your suggestions to me, jglazier@gmail.com, to Lorenzo, or to Jim Sluka. There is this Google form, which we can use also for suggestions. Please do help us develop the seminar series for now our 5th year and make sure that we have people speaking that you would like to hear. Again, if you've spoken before and it's been a while, a year or two, you have something different to talk about, you want to give us an update on your progress, don't feel that because you've spoken already, you can't speak again. This is a community and science that changes very rapidly. I'm sure that there's plenty for people here to talk about. And don't be shy about suggesting yourself as a speaker. We're happy to have people do that. Okay, we will have a steering committee meeting tomorrow at 11 a.m. To continue our discussions on the proposals to the Army Research Office for meetings on digital twins, and also for our approach to coordination with the RDA effort led by Anna Niarakis. And so if you're a member of the steering committee, please come. If you're not a member of the steering committee and you want to participate and volunteer your time, let us know and we will add you to the list. We're always happy to have people help us out. And so please remember that this actually needs to be edited. It's Eastern Standard Time, not Eastern Daylight Time. So don't show up at 11 a.m. Eastern Daylight Time or you'll miss the meeting. So I don't want to take any more time from our speaker. We have a great talk today, high-level descriptions of components of mathematical models in biology. And I will turn the floor over to our speaker.
Hello, everybody. Thanks for having me. So I'm Jan. I'm currently in Germany, in Heidelberg, working at the European Molecular Biology Laboratory, EMBL in short. And so I will be moving to Brussels actually from January onwards. So I'm now finishing my postdoc here, basically. I decided for this talk to split it in 2 parts that are actually completely unrelated. So, but I was thinking a little bit about it, and then I came up with some kind of common theme, which is this kind of high-level modeling that I wanted to discuss. So, what do I mean with this? So, I imagine that in this seminar series, mechanistic and data-driven modeling has been discussed. I imagine so. So Of course, we have these two sides. We either can start building a model for some biological process we're interested in from the bottom up by writing down, for example, a set of ODEs that describes what we know about the mechanisms going on in the system, for example, chemical reactions, gene interactions, and so on. Or we could, from the other side, if we have lots of data, but we don't necessarily know the details of the mechanism, we can use that data to also produce a model to make predictions. Now, what I will be talking about is a little bit in between, and I'm not really sure, I wasn't really sure how to call it actually. But what if we know maybe a part of the mechanism, or we know some of the important variables? Maybe we know that there is two proteins that really matter, and we know that they somehow one activates the other, for example, but we don't really know the details of everything else that's going on between these two things, so the rest of the network. Or, for example, we have some populations of cells, and we know that these cells are interacting with each other, signaling to each other, and so on, but we don't actually know what the mechanism of the signaling is. Can we still build a model, and what can we do? So this is a little bit the general theme. I will first discuss something related to bistable switches. And this is something we did a couple of years ago. This was part of my PhD. And then in the second part, I will be discussing current ongoing work about pattern formation in spatial systems. In the first part, I will focus more, would like to focus mostly on the method. And the second part, there's also, I guess, a little bit more biological questions we address there. Okay, so let me get started with part one. So this is from a couple of years ago already with Lendert Gillens, with whom I did my PhD, and the paper is out there. I would also like to mention, and this is actually the reason I think why I was asked to present here in the seminar series, that this model, we submitted it for something called Model of the Year, by this website BioModels that I have here. So this is the website BioModels. Maybe you know it, maybe you don't, but this is some kind of database where you can submit your mathematical model of your favorite biological system. It's curated, and you can search the database, look for models of systems you're interested in. And so they have this competition Model of the Year. What I'm going to discuss won last year. This year's competition is ongoing, and I do think they will also soon have a call for new entries. So if that's something you're interested in, this is a little bit of publicity for that initiative, which really aims to enhance also reproducibility in these systems and computational biology field. Okay. So let me talk a little bit about bistability. I imagine that this is something that many of you are familiar with. The classical example is a little ball in some kind of domain or terrain, like or potential, if you will. And every valley is a stable state, because if you push the ball a little bit, it will go back there. Now, I'm interested in, or what we'll be talking about a little bit, is what happens if, for some reason, I hope the video plays well, for some reason this domain is changing over time, such that the original stable state disappears and the little ball is forced to go into the other one, right? So this animation, I will restart it soon. And what you see in the right panel is, on the y-axis, actually the position of the ball, and then some external parameter that is driving the changes in this potential on the x-axis. Now, what happens there is, okay, if it reaches this threshold, the ball quickly and also irreversibly transitions to the other stable state. Irreversibly, because initially, if the parameter would go back, the ball will stay on the right. So it has to complete the whole loop, this hysteresis effect in going back. Now, this mechanism, or this switching between stable states, appears in biological systems, and the main example I want to talk about is the cell cycle. And this was also the main, let's say, biological application that we were thinking about when we were studying this. So these irreversible switches, they are used by cells to go, for example, from S phase into mitosis. So between different phases of the cell division cycle. And it's good that this is irreversible, for example, because you don't want to start, for example, segregating chromosomes and these things before, well, you don't want to fall back to the previous phase while already doing that, if you're a cell, I imagine. So it's not terribly important for the purpose of what I want to say, what the names of the proteins are. However, I will maybe be talking about this protein cyclin B, which is really driving the cell cycle, especially the early embryonic cell cycle, which activates something else called CDK1, a kinase, also called the master regulator of mitosis. And this happens in such a threshold way, irreversible switching way. Now, this is already has been known for some time, and they actually measured the hysteresis. So this is a plot from Jim Furrell's group, where if you go up, increasing the cyclin concentration, there is a threshold at which the activity of CDK1 also jumps up. But if you go the other way around, then this threshold is different. So here you have this hysteresis effect very clearly measured. Also, what causes this, this bistability and this switching, the underlying reactions are quite well characterized. So we have these different feedback loops, positive feedback loop, double positive feedback loop, CD25, and this double negative feedback loop. Okay, so this is kind of well characterized. Now, in the cell division cycle, it's cycling, right? So it's going around and around. So if you couple such a bistable switching system with a negative feedback, so in the case of the cell cycle, this would be this extra loop with APC, you can turn the switching into an oscillator, and this looks a little bit like this, right? So this is an embryo of Xenopus frog that I kind of synchronized with the simulation here. So you have these transitions between the two phases of this embryonic cell cycle, interphase and mitosis, going around these S-shaped bistable response curve. Okay, so this is, let's say, the motivation for what we were then doing. So this is well characterized. So here we know the interactions leading to the bistability, but now the question is, what if we don't actually know the reaction network, the proteins, for example, and the feedback loops that lead to this bistability, but we still want to, for example, in such an oscillating system, we would like to include this in a model. And so this is, let's say, the method or the mathematical technique, let's say, that we developed to do something like that. Okay, so it is mostly based on some kind of, well, maybe a little bit of a mathematical trick, to be honest. So the idea would be like this. If you have an input X and an output Y, so the blue line would be, if this is a very steep response, for example, modeled by a hill curve, we would actually like to kind of pull on this steep response to make it S-shaped. That's the underlying idea. Now, of course, that is not directly possible since the red line here is not a function. It has multiple output values for a single input. So we used a trick. We started from a steep response with this hill function, and then we inverted it. So we wrote x as a function of y. And then this you can actually turn into this S shape, because x as a function of y, this is fine. So we multiply this with some other function called xi here, xi of y. And then we say, okay, and then we just invert it back, and then we have our S-shaped curve. This is all very nice. But the only problem is that, well, it's not a function, and the output y actually appears there in this threshold of this hill function. However, it's not necessarily a problem. So the equation here on the left shows you how you can get around that. If you turn this function, which is not a function, into an ODE differential equation, such that the steady state here of this equation forms this S shape, then it's also good. And you can make this very fast, for example, if you include some time scale epsilon. So what we have here is some kind of module that takes input X and output Y, and the response has this bistable S-shaped curve. It's a modified Hill function, so it starts at 0, it asymptotes to 1 or to another level, and you can actually modify the shape, the thresholds, and so on quite easily by playing around with this function psi that you multiply it with. I have a couple of panels here that show this, and that show this there. So the idea here is if you have, for example, you have measured the response going up and down of, for example, two proteins, and you see, okay, there is hysteresis, it is probably an S-shaped response underlying this, but we don't actually know how to model that using a detailed model. What can we do? Okay, so two examples of this approach. The first one is actually model reduction example. So what we did there is we took model based on mass action kinetics with 5 ODEs, and we turned it into a 2 ODE model by replacing a part of the interaction network by this bistable S-shaped module that I showed you before. If you do this and then you simulate the full model, which is an oscillator in this case, then you get very similar behavior as when you were simulating the original 5 ODE model. So this is one way of reducing the complexity. Of course, there you have to make some assumptions in the sense that you need some timescale separation. For example, that all the reactions that lead to this S-shaped curve should be happening faster than the rest of the system. So this is one example. The next example is we try to model the somatic cell cycle, which has more complexity than the early embryonic cell cycle. And we did this by linking different bistable modules in a chain, where the output of one becomes the input of the next one, or maybe indirectly. And in this way, you can have, for example, things like this, where you have 3 bistable switches, and in this way, you can model transitions between different phases of the cell cycle, a more extended version of it, basically. This is the idea here. So each of these S-shaped response curves is explicitly put into the model. So you don't need that many equations to get this behavior out of it. And then you can use this for different things. For example, one thing we did is, well, for example, the cell cycle can be influenced by the circadian rhythms. And maybe it's not exactly known how the well, on which protein or which gene it affects, but you can say, well, what if the circadian clock actually modifies the threshold of one of these switches, right? You can directly include that in this model without having to go through the chemical reaction network. Okay, so yeah, this is the method we proposed. I actually would like to mention that I think it may have more broader applications in the sense that you can build a network of these bistable input-output response curves and link them up in any way you wish, right? The input of one can become the output of another, but you could also say the output of one could modify the threshold of one of the others, for example. And it may have some applications in other fields, such as climate dynamics. So I took this figure here from a recent review paper, and in climate studies, they often talk about these tipping points. For example, the melting of an ice sheet. This is quick and irreversible, at least in the short term, because it takes a long time to build up an ice sheet. So this is also this hysteresis behavior. But then there are these connections between different tipping points on the planet, where if one thing tips or switches abruptly, it can affect other systems on the Earth. And people have actually already been modeling these things and looking at cascades and so on, also with bistable modules. But I think this might be a potential application for it. And of course, I would be very happy if any of you have some idea, for example, of using this. Okay, and this actually brings me to the end of this first part. Now I will switch to something completely different, which is pattern formation, which is what I'm currently working on. So here in Heidelberg, I'm of course not alone, and I will be showing you some experimental data. And here I think it's very important to acknowledge Michael, my friend and colleague who did all of the experiments. And throughout this whole project, we had a very nice interaction between the theory and the experiments, let's say, where from the beginning of the project, we combined both to go forward. Okay. So pattern formation, I imagine that many of you are familiar with it. Animal coat patterns is one of the obvious examples. So these are regularly repeating spatial patterns made up by much smaller, typically individual units, molecule cells, things like that. The interactions between these units that lead to the pattern can be chemical, reaction diffusion type systems, but they could also be mechanical. And one example, for example, on here on this slide is the bottom left picture. So this is a bacterial biofilm, which is growing on a substrate, and the substrate, well, it doesn't grow with the biofilm. So stress builds up until it buckles, just like you would have a sheet of paper and you push it together, it buckles. So this biofilm buckles and it forms this regular spoke-like pattern when it buckles. So this is an example of a mechanical pattern, so to say. Also, of course, in the studies of pattern formation, since the work by Turing, mathematical modeling has always played quite an important role in this. Now, the patterns that I showed you here are relatively large scale in the sense that you have many repeated features that make up the pattern. However, the same mechanisms and feedbacks that lead to this pattern formation are also important on much smaller scales, I would say, and they are important in the early development of organisms. So here I have this video with C. elegans embryo, I think a single cell stage. And here you see a pattern forms that basically gives a half-half type pattern. Like so the cell, it polarizes like here. The movie on the right, this is what is called a gastroloid. So this is an in vitro system, and it's basically a ball of stem cells, a couple of hundred of mouse stem cells. And you see at some point, half of these cells, they light up green. So they start expressing some gene. Importantly, in the beginning, it was just a homogeneous, uniform ball of cells, so to say. It breaks symmetry. One half starts to express this gene, and it also grows from that side. Right? So this recapitulates some of the things that are happening in early development. And these symmetry breaking events, so they're due to cell-cell interactions, for example. And it's very important for the initial establishment of body axis. So like a head to tail. At some point, if you start out from being a homogeneous uniform ball of cells, you need to establish the head-tail axis, for example. And these similar mechanisms that lead to maybe these stripes and so on could also lead on small domains to symmetry breaking. Okay, so what are we really interested in? So, of course, the general idea is, well, how are these patterns formed by cell-cell interactions? But we're particularly interested in this aspect of size and shape of the domain on which these cells can move around, for example, how that controls or affects the patterning. And in the end, we would also like to learn something about development of these organisms. Okay, I will first tell you a little bit about the modeling approach that we're using. So here is the equation. It's a relatively simple equation in the sense that there is only one variable. So a density of cells, how many cells are where in time and space. And the only thing that can happen here is cells move around. So there is no cell division, no birth, no death, and nothing else. And cells can move around either do random motion, diffusion line, this is the yellow term, or importantly, there is an advective term in this equation. And this models cell-cell interactions. So attraction, for example. And this is so you see the integral, this is a non-local term that we use to model the fact that actually the cells can interact over some spatial range. It's not local. this is a relatively generic model. And models like this are called non-local advection diffusion models. They have been used already actually since quite some time, but more intensively, I have the impression in the last 15, 20 years. And what is quite nice about them is that it doesn't really matter if you're modeling cells, but you can also model animal groups, for example. So the same equation has been used for multicellular systems, but also for animal herds and flocks and these kind of things. And related to my, let's say, my general theme, you don't really need to know the interaction mechanism. So how these cells are being attracted or repelled by each other. You need to prescribe this using a function that says how strongly are they interacting as function of their distance. Like the one here on the bottom left. I'm usually working with exponential decay with a certain length scale. Okay. Now, in principle, it would still be interesting to know what mechanisms could lead to which kind of interactions. So here is just to give you an idea. Maybe the cells are secreting some chemical, which is then sensed by the other cells by which they want to move together. It could be something related to cell-cell adhesion, or the cells sometimes they extend protrusions for signaling, which could also lead to a long-range interaction. It's not entirely clear for each of these mechanisms what is the best effective interaction function, for example, to use. However, we don't need to know the mechanism per se. In principle, we could estimate this directly from the data. So again, a bit in the similar vein as what I said about this S-shaped bistable responses, that you could basically use data to say this is my response. Here you could use cell tracks, for example, to infer or estimate what is a cell-cell interaction kernel. And this is also something that we are doing. However, it's not so easy in practice. So in theory, it should be possible, but of course, these systems are very, very noisy. Good. I've been talking a little bit about the theory. Now I will tell you a little bit about, well, I have given the setup of the theory. I haven't really shown any results, but first I would like to tell you a little bit about experiments that Michael is doing. So Michael does experiments with primary cells, so from mouse embryos. He takes the cells from the embryo and then he randomizes them. He mechanically kind of mixes them around so that the cells, let's say, forgot where they were or what the neighbors were, so to say. So they start out from a well-mixed initial condition. Michael then puts these cells in wells or on micro patterns, so on certain shapes. And what happens is that, well, the cells form patterns. So on the left, I show you a movie on a relatively large domain. I hope you can see it well. Yeah, so very clearly see these spots forming. So I should mention that what you see is, if anybody is familiar with it, so it's the intensity of the image correlates with the expression of a gene called brachyury, which is involved in these symmetry breaking mechanisms in early development. Also in a marker for the mesoderm. But it's not really important for what I'm saying because these bright spots, they also correlate with high cell density. So there are just more cells also in these bright spots. Okay. On the right is a simulation of this equation that I showed you. So this is on a similar spatial scale using relatively straightforward values for the parameters. And you actually, in these types of models, you get nice patterns almost for free in a way. If your cell-cell interaction strength overcomes the random motion of the cells, you will see these patterns forming usually. And already, well, you have to do minimal tweaking, so to say, to get something that looks like the experiment in this case. Okay, so these models, they can form patterns. That's relatively straightforward. But what we're really interested in is this question of, well, if we now look at what is the effect of the size and the shape of the domain on which these cells can move, right? If you restrict that, because this was a relatively large domain, we can use the mathematics of infinite domain if we want, and that works fine. However, how do we now study bounded domains. And here we went to the simplest thing, which is one-dimensional domains. And in one dimension, shape is basically reduced to the length of an interval, let's say, of a one-dimensional thing. And for this, Michael actually managed to perform quite a lot of experiments looking at this. So what he did is he printed these thin bars coated with fibronectin, so the cells can only stick to these predefined shapes that we choose, basically. And he printed multiple lengths, so different lengths. I'll show you one example here, but we have a bit less than 300 in total of different lengths. You also see it doesn't always work out very nicely. Sometimes these cells, they try to kind of bridge, make bridges and so on, but we have quite a lot of them that we can use. So these cells are moving around, they're forming these patterns, but they're restricted to certain types of domains. Here you can see a cropped out bar. So these cells are all moving around. And in the end, they form a single spot on one side. So this is an example where it actually forms this half-half type pattern. And the bottom video is the density of the bright cells, I should say. So the cells with the highest intensity that are expressing this gene, because those are the ones that are actually interacting with each other. And so we have a pipeline of processing the images to these one-dimensional profiles. And another thing we can do with these bars is we can make chymographs, or so space-time plots, basically. So if we squish the bar along its short axis and then plot time and then the intensity in space, we get this. So here you clearly see that after a long time, well, 1,000 minutes is I think is the cutoff. So these experiments, they run for 15, 16 hours normally. So these cells, they form this pattern, which in this case consists of a half-half type pattern. Okay, so we want to understand this. We also want to know in the model what happens. Here I will tell you a little, slightly little bit of the mathematics of it. So the model on a bounded domain, this would be the new equation where I included now something extra, which is effect from the boundary. This is to make the model a little bit more general. It could also be zero, this term, but so we allow for the fact that maybe near the edges of these bars, cells, either they want to be there or they don't want to be there. So we allow for some interaction there. Okay, so we have this equation, we have this model. Let's attack it. Let's kick it and see what comes out of it. The first thing you usually do is something called linear stability analysis. Maybe you're familiar with it. If not, very quickly. Basically, what we do is say, okay, let's assume that we start out close to some uniform profile. Is this stable or unstable to perturbations? If it's unstable, it means that perturbations can grow in time and that a pattern can develop. So let me start it here again. So this type of analysis tells you two things. First, under which conditions on the parameters of this model do you get this pattern? But it also tells you what does the growing pattern look like? Which spatial modes will grow? The classical approach is to expand your perturbation into Fourier modes, sines and cosines, and then look basically which wavelengths will grow in time. Very briefly, even though it was kind of a lot of calculations, in our system, Fourier modes don't work anymore. Basically because you have a problem with this integral and then the boundedness. Basically, you break this translational symmetry. So, however, we could do the calculation analytically in some cases. But what we find is that the spatial modes that you need to describe this linear stability, they actually depend on the parameters of the system itself. So I won't go into the details, but here you can see that the spatial modes that you need, they look a little bit like cosines, but they're not exactly cosines. Oh, okay. That's just if you're interested in those kind of things, we solved this integral differential eigenvalue problem. Yeah. Anyway. We also see that there is only one parameter really that matters for this pattern formation, which is this alpha bar, which combines parameters related to the cell-cell interaction strength and the random motion and the density. It depends only on that, and of course on the length of the bar as well. Another thing this analysis tells us is that, okay, when do patterns form? Basically, if the bar is long enough and the cells attract each other strongly enough, you get patterns, which in some sense is a generic prediction. For you would probably get this from other types of models as well. Right? Okay, so then we went to the data and asked, okay, do we actually see anything like this? Is this a good description? So we asked the data directly, what are the modes that describe the patterning? And we used a principal component analysis. So plugging in these density profiles over time, we asked what explains the pattern growth. And what falls out is actually modes that are very similar to these modes you would find in the model. Moreover, we also found that on the longer bars, you need also the higher mode. So you have more, it's more likely to form multiple bumps, for example, multiple aggregates in the patterning regime on the longer bars, which is expected, but it does show you that the model and the and the pattern formation mechanism that we see in this biological system really is what we think it is in the sense that it is this instability-driven pattern. Okay, so this is one thing we found. The other thing is then not initial pattern formation, but what happens after a very long time. So we looked at the steady states of the model, of the full equation, so the full nonlinear equation. We tracked the density profile, the steady states, as function of the parameters. And here the main message is that for a wide range of parameter values, you actually expect it to polarize for all the cells to end up on one side. Unless you have very strong interactions with the boundary being either attractive or repelling, but for a wide range of boundary interactions, you expect it to polarize, even if initially it formed multiple bumps. And then we also tracked, like, did a 2-parameter bifurcation diagram to track these pitchfork bifurcations. That's a technical aspect. Okay, this we also see in the data on many occasions. For example, here initially you find 2 aggregates that form, that coarsen, and they join up into one, which is one side. And this happens a lot, but not always. So I have to be honest here. And sometimes we see this. So here, actually, it forms 2 and it stays 2. And here it forms 1, but it is in the middle, not at the side. So this I'm still thinking about. Can we either, well, we could reconcile it with the model by just saying, okay, these are transients and it hasn't reached any kind of steady state. But yeah, maybe there's a little bit more to it. This is, well, to be seen. However, on average, if you define some polarization score and you look how do the bars do, the longer you wait, the more polarized they become. So that seems to be on average, even though the effect is not super strong, also because in these averages, you also get these things, for example, right? So, but the polarization seems to be mostly there. Good, and I think this is the last thing I wanted to tell you about this second part. So let me conclude this. These types of models, these non-local advection diffusion models, are very effective at describing pattern formation. You can also generalize them to many different cell types and populations. I only used one because it's the simplest, but in principle, you can have many cell types and define interaction functions pairwise for how each type affects each other type. And this, in principle, you could also estimate from data. Then one of the main messages here is that the boundaries do matter and that the short time versus long time difference is there. And then finally, yeah, we're also still working on this. And one thing we're doing is 2 dimensions, basically. So Michael has this discs that he printed. So we're also analyzing that. And of course, it's estimation of this interaction function, which is, well, still ongoing. With this, I would like to thank you for your attention. Any questions, of course, welcome.
Okay, please. Great talk. Please, questions from our audience. Holly, did you have a question?
Yes, I did. First of all, thank you for your talk. It was really interesting. mean, I have a few questions actually, but I guess I'll start with the one. So I was curious to know if you were, when comparing simulated patterns to the experimental patterns, if you were quantifying the pattern at all in order to compare the results, or if it was just a qualitative analysis.
Are you referring to this big domain, this one, for example?
Yes.
Yes. So no, so this experiment is not super easy. So we have three repeats of this. And so Michael actually, what he did is do a 2D Fourier transform and look at the growth rates of each of the spatial modes. And he has a dispersion relation. So, and this is something, I haven't done it yet actually, because this is very recent, but we could actually fit some model parameters already from this, for example. Yeah, so we're also comparing it in a bit more detail.
Lorenzo, go ahead, please.
Hi, Jan. Thank you very
Hi.
much. It was a very nice talk.
Thanks.
I have one that is probably a little bit generic. Let's say you discussed that you infer some equation that tells you a certain dynamic of your molecule of interest or cell of interest, etc. Then you say if that fits the data, then I have a good argument to convince people that, all right, that might be a good way to really model that particular system. And then it comes that very often you have, I don't know, you say you have an oscillatory mechanism and something, and you might have more than one simple way to achieve an oscillatory behavior, etc. And then might be the different models equally or somehow equally map the data. How can we do to convince then people that we chose, let's say, the right way, the right model? And it's always difficult, I know, but it's just for discussion, perhaps.
Yeah, no, I'm sure if I have any kind of definite answer to that, but I think it's a good point. Yeah, you have some, I guess, degeneracy. Depending on how much data you have, there may be many models that fit it. Ideally, you predict what would happen if you do a new experiment and then do the experiment, and then if the models give you a different prediction, that's I guess how you in the end. If you don't have the ability or to do new experiments, I'm not sure.
Sometimes it's also that we know something from the literature.
Yeah.
Because people have done experiment before. We try to create a model, but then we say, all right, let's say I can have oscillation via a positive feedback model and a negative feedback model. And then the literature tells me that probably is a negative feedback model. And then I find that my data really map better to the positive feedback model. And so it becomes a conundrum sometimes. So I think it's important to have this kind of discussion to keep it up.
Yeah, no, I agree. Yeah.
Well, thank you anyway. I really like the talk. Thanks.
Hello. Hey, and this is Chiang from Emory University. Very interesting talk, especially the first part of about the biostability there. So my understanding is that you come up with a kind of more like Hill-like solution, right? But that allow you to describe this S curve, which has a negative slope in the middle of the curve. So in one of the slides you showed you that's whatever the function that's being used to modulate the K term in the Hill function, and that can here, and that can help to I would say so that modulating function itself is a function of y, or is something
of y. It's a function of y.
Okay.
So how you can look at this actually is a Hill function, but where the threshold itself depends on the output of your Hill function somehow. So it's not a function.
Right. It's kind of like enforcing a feedback, almost like that, right? Yeah.
Yes. Yeah.
Right. So I'm curious how, let's say, because this become very useful, but sometimes I just want to have a bistable switch in my system. I don't care what actually making of that bistable switch. So I often kind of just implement something like X, Y, mutually activating each other, or maybe just Y autoregulation. Right. So I always kind of struggle a little bit. Like I want to define the switch, have a defined bistable zone for the on threshold versus the off threshold, and also have a defined off state versus the on state. Right. Basically, you want to have a define your zone both for the Y and also for the X. So with this approach, you can just fine tuning those easily. Like say in one of your thing there, I can fine tune where my two threshold is, and I can fine tune where my off level, on level there.
Yeah. So
Uh-huh.
this is what we did here. So we
Mhm.
had the middle panel, we had this gray one,
Yeah.
and we basically chose this modifying psi function to have thresholds at the same levels. And then the green one is then the approximation we used. So the zone in which you have the biostability is, you choose it. You can control that. You can tune it.
Okay. Can I also control in this case how much, say, your lower limb, your lower branch, kind of rising from 0 all the way up to the threshold point?
Yeah,
Uh,
you can. So okay, so if I remember correctly from the paper we did,
Yeah.
you can control for the threshold points both the horizontal and the vertical position.
Right. Okay, I see.
Yeah.
And then if you want to have a kind of a inhibitory bistable switch where the increase in the inputs leads to decrease in the outputs, you can just
I guess you can just flip it. Yeah, we actually
flip.
didn't do that
Okay.
explicitly, but I imagine it's
Yeah.
just flipping it.
Okay. Yeah. Thank you. Very nice. Yeah. Thank you.
Yeah, thanks.
I wondered maybe if you could talk a little bit more about this question of designing maximally discriminatory experiments. I mean, here you have mass action versus piecewise. It might not be so easy to develop an experiment to distinguish those two. But
Yeah, I
in
agree.
the pattern formation case, for example, the Miggenson paper has this discussion about the possible types of interaction, and maybe some of them are going to depend on the diffusion constant, some will depend on the interaction, the filopodial length, and so on.
Yes.
Can you talk a little bit maybe about some concepts that one could use in your methodology that might help people design effectively discriminatory experiments to disambiguate these things that
Yes, it is a good point. And well, I have a backup slide, but actually showing that it's difficult because where is it? Here, this one. So these are two different, if you will, mechanistic models. So the left one is a chemotaxis, where there is a chemical produced by the cells, and the gradient of this chemical leads the cells to move around. Now you can turn this into this non-local version if you assume the chemical is fastly like equilibrating. And you get an exponentially decaying kernel where the length scale depends on diffusion and degradation of this chemical. The right-hand side is an example of a mechanical active matter type system where cells contract, pulling in more cells. And here you can also write it in a non-local way, and you get the same non-local equation also with an exponential kernel. In this case, the length scale, however, depends on, so this is hydrodynamic length scale, depends on viscosity and friction. But they give you the same the same interaction function with completely different mechanisms. So this shows you that, well, that the question is difficult at the very least. However, of course, the length scale should depend on these parameters. So if you have access to perturbing the chemical that you think might be playing a role, and you can perturb that, and you see that the length scale changes, and the patterning length scale itself depends mostly on the interaction length scale. This would be a way of getting at it. And if you perturb the chemical and nothing happens, okay, then maybe it's not that chemical that is doing the interaction. Yeah. So that's what I can say about this.
That's an interesting one. We ran into that when we were working with angiogenesis, where you have the hypothesis that you have cells, the endothelial cells secrete short diffusing VEGF, and they chemotax to that VEGF field.
Mm-hmm.
So you have essentially a autocrine / paracrine chemotaxis. If, on the other hand, you assume that the cells put out filopodia and pull on each other, you get exactly the same mathematical form.
Mm-hmm.
All that
Yeah.
matters is that you have an attractive interaction that decays over a short range. And so that could actually be filopodial interaction between the cells directly. It could be a chemotactic interaction, or it could be an interaction that was mediated through the extracellular matrix. All of those would give the same mathematical form. As you point out, the particular parameters that they depend on will be quite different. In one case, it'll be the diffusion constant. In the other case, it'll be the length of the filopodia. In a third case, it'll be the stiffness of the extracellular matrix. But I think this, I think, also is a nice illustration of why it's so important to be careful about writing these control parameters in an intelligent way. Because I think mathematically, we often write those differential equations down at the top and say, well, this is the structure of the differential equation, and then we don't really think perhaps always about what the critical parameters that lead to the bifurcations are, or here are the length scales. So I appreciate your comments on that very much.
No, thanks.
The other question I had, and I should turn it over to other people if other people have questions. I always I don't like to monopolize the floor. Was that here you're starting essentially with these continuum models, these reaction diffusion or advection models. A lot of people in the room do agent-based modeling. Now, I was wondering if you could comment maybe a little bit about how you might apply some of these concepts in an agent-based modeling context. Presumably, the interactions between your agents would look like the interaction kernel in some way.
Yes,
how would you do
depending
this?
a little bit on the details, exactly that, I think. So you can actually, I think you can coarse grain an interacting particle system into and get almost the same continuum equation as what I showed you there. So I did play a little bit with agent-based simulations at some point, and well, the behavior is relatively similar to the continuum limit. Yes, so I started out with continuum equations, but coming from maybe a mathematics background, I wanted to try and get some analytical feeling for it. So that's maybe one of the reasons. I'm not sure if it's the best reason, but like a personal reason. However, I think it's important to ideally you get the same result using different modeling approaches, and then.
Well, we certainly would like to be able to define the interactions between our agents based on experimental data. So the basic methodologies and concepts, I think, are quite important.
Yes, so there are estimating this interaction from the data. We could also do this in two ways. In principle, you could try to fit a continuum model directly if you heavily smooth your data. For example, it's not so easy. I think I haven't really tried it really. Or we have cell tracks and we can go from that. I don't actually know which one is easier. It's actually a good thing to figure out, yeah.
Other questions? Feel free to chime in if you have questions.
Something I wanted to ask about the final outcome with favor that the bars over time will polarize. I was wondering if you had a biological hypothesis for why that's happening, like because it clearly emerges from the mathematical model. Like, was one of those mechanisms included in your cell interaction equation found to be more sensitive, or if the result was more sensitive to maybe one of those, or what do you think might be going on biologically?
Biologically, yeah, that's a good question. So the generic answer is that just having boundaries leads to this this polarization in the end. It's just the fact that you're bounding your domain and something has to happen. And okay, polarization seems to be in this case. Now, an aspect that is, well, maybe it's also a mathematical answer, is that these longer range interactions, if you feel the boundaries, you feel how much space there is between you and the boundary, and this leads to the whole thing moving in one direction. I don't think I can give a very intelligent biological answer to your question. Yeah, it's a bit confusing in the sense that the biology does sometimes other things also than the mathematics, of course. That sometimes these cells actually stay in the middle. So this is a little bit of a puzzle still. Michael tells me that, okay, we're using a heavily simplified mathematical as well, right? So these cells, there may actually be slightly different cell types in the mix that do slightly different things, and we're just saying there's one thing that explains everything. Yes, but so I don't know is the short answer.
Well, I'd like to thank you again for coming. I know it's late in Europe, so we appreciate it very much. Where are you going to be in Brussels?
So I will be at the ULB. I will join the group of Geneviève Dupont. Maybe that name rings a bell, and Didier Gons, and they work on oscillators mainly.
Okay, well,
Chronobiology, yeah. But I will keep on working on these pattern formation models, and I will hope to kind of broaden a little bit the applications.
okay, well, thank you very much for a very stimulating talk, and I look forward to hearing you maybe come again and update us on your progress.
Thanks for having me. It was fun.